Okay, everyone. Hello. This is Group 8 for Engineering Ethics 316G. Our presentation today is going to be on gene editing, a sort of ethical review of this new sort of developing technology, its applications, and its impact morally in the world of technology and science. My name is Steve Sullivan.
My name is Jared Hill.
My name is Vanessa Lemoyne.
Jackson Eisen.
Carolina Shinowski.
Mitchell Farah.
Okay, so what is gene editing? Gene editing essentially involves the altering of segments of DNA within an organism in a very precise or targeted manner. Often the purposes behind this practice is to correct genetic defects or add beneficial traits to an organism, and in general study gene functioning. Modern applications include agriculture, enhanced research in biology and medicine, and there is definitely potential application for use in environmental conservation. So far, there are a couple of companies that are pursuing this sort of thing, namely CRISPR Therapeutics, which was formulated in 2013, and they develop gene therapy services in clinical trials, which are prospective biomedical research studies. This gene-based medicine has potential to offer a solution to those suffering with chronic diseases and all sorts of other applications, and in fact has been used already in the past. So this technology is very real and in our world.
All right, so to get us up to date on the gene editing world, we'll do a little current state of development. Rapid development has emerged since the 1980s when scientists first harnessed the bacteria to split the double helix. Since then, numerous methods have arisen, including zinc finger nucleus, transcription activator-like effector nucleases, and CRISPR. Each of these methods have benefits and drawbacks that make them useful for different reasons. And these developments have led to a deeper understanding of the human genome. Gene editing is also being used in aquaculture and agricultural settings, with commonly targeted traits being reproduction and development, as well as growth. Additionally, special gene markers such as trans-GFP may be manipulated, as well as the omega-3 metabolism compound. Further gene editing therapeutic studies are being conducted for medical development purposes. These technologies are also invaluable in diagnosis. In recent times, CRISPR in particular was used during the COVID pandemic to detect COVID. At the beginning of the pandemic, the detection of COVID was slow and often inaccurate as COVID mutated rapidly. By using CRISPR, researchers were able to identify common sets of DNA among most strains, and these common sets of DNA would then be compared to a test sample to determine if one had an infection or not. This led to highly accurate and quick test results without the need of highly trained technicians or expensive labs to conduct traditional tests. Additionally, gene editing is also useful for treatments. There are currently over 70 different clinical trials involving gene therapy to treat cancers being conducted by the NIH. These trials have been make use of various different methods of gene editing. It's commonly being used to treat infections such as HIV, HPV, herpes, and the coronavirus. It's also being used to address genetic disorders, including metabolic, neurodegenerative, and ocular disorders. Further, it's being used to treat cancers like leukemia, lymphoma, and melanoma. Also, it's being used for treatment in genetic diseases. Each of these use different methods of gene editing, and the benefits emphasize the diverse applications of gene editing.
There's a lot of further research and development to be done for safety assurance of gene editing, and more specifically regarding the CRISPR technology, there are four main areas of concern. The first of which being the delivery challenges. They're delivered in a variety of approaches, such as viral, physical, and extracellular based systems. The issue with having so many options to choose from is choosing the right one that'll cover all the bases. There are multiple technology is already being developed using a combination of delivery systems or systems such as viral or non-viral delivery systems. The next area that needs development is the off-targeting, which occurs when the Cas9 protein brings the gRNA to the wrong area due to too many gene similarities, and it can cause further mutation and adverse effects that you wouldn't expect. And they are developing more bioinformatic tools to more accurately predict these areas, and also through the use of Cas9 nicases and adding anti-CRISPR proteins to inactivate these proteins after they are already have targeted where they need to be. It reduces the bad effects. The dysregulation of multiple genes often leads to cancer, which emphasizes the fact that gene polymorphization must be observed. In order for the system to be affected, the proper gRNA must be designed to target and prevent interaction and progression of cancer. The bioinformatic tools are also used in order to regulate these genes. Lastly, the autoimmune responses that can be caused by CRISPR need to be further researched, because when the body recognizes these proteins as foreign, it can develop a reaction that will prevent it from actually performing the gene function. Research has currently found that by using it earlier in the lifetime, it's more likely to help. And in addition to this, by targeting the immune privileged organs, it can allow the body to accept these proteins. There are also ethical concerns for safety, such as unintended consequences that will cause further harm because of these scientific challenges that I mentioned earlier. There's also been a concern of inadequate access, since it is very costly and needs a lot of procedure over time. In addition to this, people are also concerned that it'll disrupt your ecosystem, and there are also concerns of designer babies, which are where people modify the embryos and make enhancements to them, and they pose a threat of an unlevel playing field, and they also causes a loss of a sense of normalcy. Very recently, the FDA has approved gene therapies to treat patients with sickle cell disease. This represents a very important medical advancement for the future of gene editing technologies, and shows that it is determined to provide more benefits than risks. And this research will help offer new avenues for treatment, so that as more patients are being treated, more data can be collected.
So we can look at trust issues with gene editing and the public's perception of the technology. So ethical concerns of gene editing includes morality considerations and stakeholder impacts. One ongoing debate pertaining to the technology is human rights and autonomy, particularly for developing embryos. There is also a considerable environmental concern, as GMOs may distribute human-altered genes to organic or non-edited wild species. On one hand, altering genes, for example, the American chestnut tree, to be resistant to foreign blights could bring it back to its full robustness as a giant North American shade tree. However, the genes of the American chestnut will then be forever altered, and those alterations will impact its evolution and impact the environment in which the species resides, in addition to other species that come into contact with it. So media coverage may experience inaccurate reporting, which can lead to misinformation being spread about the technology and misconceptions developing, all of which hinders trust building with the public about GMOs. And some surveys were done on the usage of gene editing in the United States, and the majority of people surveyed agreed that treating a baby's serious disease could be an appropriate application of gene editing. However, overall, many Americans only like the idea of gene therapy versus altering genes to potentially make a child smarter or more attractive and just edit the embryo in the womb. And there is some resistance to the usage of the technology in agriculture as well. So this image is from the New York Times, and it shows a genetically modified tomato developed by the prestigious botanist Kathy Martin. And this tomato has superior traits, including a higher nutritional capacity compared to non-GMO tomatoes. Martin's GMO breakthrough has anti-cancer and anti-inflammatory properties, as seen in studies with mice who live 30% longer than mice fed regular tomatoes. So this is just one example of a positive application of GMOs and it being represented in a positive manner in the media.
So further trust issues with gene editing, we have challenges and concerns by that. So since gene editing technology has gained momentum in the past few decades, trust issues from the public have arisen, including concern for environmental impacts, autonomy and consent, particularly of human embryos, accessibility, and controversial introductions. Germline cells passed on genomes from one generation to the next. Hereditary changes will have a lasting impact on future generations. Changes in the genomic makeup of GMOs may be present during gene flow, during reproduction in both target and non-target organisms, resulting in contamination of organic species. Adverse effects include increased pesticide resistance, decreased biodiversity, and the development of hyper-robust species. For autonomy and consent, it is a concern pertaining to developing embryos in particular. Which is a question that arises, is who is right to make these gene editing changes. How ethical is the action depends upon the purpose. For example, the difference between eye color and a genetic disease. Another reason, accessibility to different social groups, as well as in crops. GMOs have been highly controversial in crops and agriculture. Sometimes an advancement of the technology has been made public and initially has seen great results and experienced positive public perception. However, significant drawbacks of the publication were seen later. This experience results in decreased public trust as there is a level of deception felt.
Okay, so in terms of regulatory proposals, gene editing and therapy is a very complicated and not well understood subject in the public eyes. There are essentially two different ways to edit DNA, and that is germline editing, which is the editing of base human DNA that's passed down through generations, and therefore is a lot more impactful. And then there's also somatic editing, which is the editing of mature adult DNA, which has no potential of being passed down. So the largest concerns around gene editing are edits to a human germline as they pose to be passed down through a lineage, which could pose potential problems in the public eye, as future generations would not have any say or autonomy in how their DNA was affected or changed through germline editing, which is obviously a very clear problem. On the other hand, somatic therapy is just making edits to an adult's gene that have nothing to do with potential lineage, which is preferred regarding autonomy. Internationally, there are no existing regulations to control the use of gene editing. However, in the US, there are existing regulations, which I will get into shortly. The use of germline editing is not supported in the US and will not be considered until copious studies have been conducted in non-human trials to sort of view and see the real effects of gene editing. As for current US regulations, in 1995, the Dickey-Wicker Amendment was passed that forbids the NIH from funding any clinical trials involving the manipulation of human embryos. Later on in 2015, Congress passed a provision that stated that the FDA must approve any human clinical trial involving gene editing, but they must deny any that are trials to modify the human germline. In 2019, a company by the name of Odin was selling kits that would allow regular consumers to get access to CRISPR kits. But shortly after that, California passed a law prohibiting the sales of any gene editing technology to regular customers, as the potential impacts and the lack of regulated experimentation would pose a significant hazard. As a general catch-all, the FDA does have the final say over what one can sell in the US market. So the FDA ultimately has the final authority over what is for sale in the US and potentially could override California's decision. The FDA's stance as of 2023 is that money is allowed to be used for only somatic gene therapy and not germline therapy. So there seems to be a general consensus among scientists at the moment that editing of the germline is highly risky and needs further research. And the regulations that have been proposed and enacted seem to match these ideals. Somatic gene editing and therapy are approved, and they're highly encouraged as they prove to be a very potent tool in the treatment of serious ailments, as well as many other applications and things like that, such as modifying food and GMOs and things like that. These regulations even promote the use of somatic gene therapy, leading to a rapidly developing field of science. However, germline editing is discouraged as it risks very serious adverse effects. Though this is a cautious approach and it may take a cautious approach to take, it may mean that the potential benefits of germline editing will never be achieved or will take much longer to fully mature in the field of science.
So the industry has a significant role in trust building with public perception. According to a study done on identifying public trust in building priorities in agricultural gene editing, 3 main criteria were established, resulting in a typology of gene editing trust issues. So the criteria include proponent priorities, critic priorities, and contested space. Proponents of and in the gene editing industry are driven to learn from the research and experimentation being conducted on genetically modified organisms. Industry proponents also engage in efforts to build public trust by focusing on shared values, avoiding scientific talk or non-specialized language, and maintaining communication transparency. Critics who represent advocacy and public concern would report greater concern for potential risks of gene editing applications in agriculture. They suggest improving regulatory oversight as one solution to protect and maintain public rights. Additionally, concern about GMO and organic cross-contamination is cited as an important consideration when implementing gene editing technology. In the contested space, discuss points include clearly framing benefits and risk criteria for intention of increasing the public's knowledge, and which may include product labels. Additionally, appropriate definition of gene editing is a consideration, and it's still an ongoing discussion.
Further industry roles and trust building, gene editing in crops can increase nutritional levels of the food. So if companies wanted to support their products and dismiss fears of eating genetically modified food, they should list the nutritional benefits associated with the food. Another big selling point for people can be lowered costs of the food. Altering the food can help increase how long it remains healthy to eat and how much of it is usable after being collected from, say, farms. This in turn can help lower costs because the demand for the food would be much lower, as much would be more abundant. Maintain the food's health also encompasses how the crops grow. Pesticide use can be drastically reduced by editing the genes of the crops, allowing for higher yields. Reducing the needs for pesticides also helps keep harmful products out of the soil and water. And another point would be removing potential allergens from the foods to make it more available to people. In contrast, it does pose risk to the biodiversity of plants in nature. Natural crops can be affected by the growth patterns of edited crops or plants and could outgrow them or take resources away from them. This issue can be attributed and similar to that of invasive species problems we see today.
Two very important subject matter experts in this field are Emmanuel Carpentier and Jennifer Doudna, who together developed the CRISPR technology. Their research culminated in an easy and precise way of changing any organism's DNA. Both Emmanuel and Jennifer believe their research will enable people to alter diseases that cause genetic faults, leading to improved quality of life for people. This thought that their work improves life provides them with a bias. They want to continue pushing for their work's advocacy. Given that, another example is Kathy Martin, who works in biofortification, which is enhancing the nutrients in food by performing metabolic engineering to enhance phytonutrients, which are compounds that provide health benefits. People surveyed in the United States believe almost the opposite of what these gene-altering subject matter experts believe, which should be taken into great consideration. Most people would not want to alter their baby's genes because they don't have the ability to consent to the gene altering, as well as the risks of adverse side effects or the disease that's being targeted not actually getting fixed. A minority of subject matter experts in the field believe that gene editing should be used as a last resort, which Jennifer Doudna, while being a proponent of gene editing, was quoted as saying that there is an urgent need to confine the use of gene editing in human embryos to settings where there's a clear unmet medical need and where there's no alternative viable approach, which was in response to Dr. He Jiankui at the Southern University of Science and Technology in Shenzhen, China, utilizing the CRISPR technology to alter the genes of twin baby girls before they were born to prevent them from being able to develop HIV, as their father was HIV positive. And he did not follow the proper protocols, and he did not attain approval from the directors at his organization to do so. So there are many cultural and generational considerations pertaining to gene editing technology that should be discussed. Some diseases, such as sickle cell anemia, affect certain races more than others. Sickle cell anemia is a debilitating, lifelong disease, including chronic pain and compromisation of both mental and bodily health. It is more likely to affect African Americans than any other racial group in the United States. According to the Cleveland Clinic, about 1 in 12 people of African American descent carry the sickle cell trait. African Americans have been the subject of highly unethical experimental medical treatments in the United States in the past, including an experiment led by the University of Cincinnati, during which over 90 African American terminal cancer patients were unwittingly exposed to extreme levels of radiation without their consent. This isn't the only famous incident of American medical professionals experimenting on African Americans. And experiments of this nature give more than enough reason for distrust in new experimental medical technologies from American people of color. Another famous example of blatant racist practice in the American medical field would be the Tuskegee syphilis study. Studies like these are more than sufficient reason for American people of color to mistrust new medical technologies, including CRISPR gene editing.
So, as discussed, we see that gene editing has helped alter segments of DNA through specific targeting, especially since the 1980s. Developments in research have led to deeper understandings of the human genome, treatments to multiple infections, over 70 clinical trials for cancers, and agricultural improvements. It has since been approved by the FDA, but there still remains significant controversy and public concern over embryo autonomy, hereditary impacts, and the environmental impacts they could potentially cause.